Synaptics. All right. Can everybody hear me at home?
Yes.
Yeah.
Okay. Good. All right. All right, let's do this. Okay, well, welcome back to the semester such as it is. We'll try to pick up where we left off and make a little progress. There are enough of you who are dealing with enough stuff, but I think I'm just gonna send the link to everybody. And I think that actually I've got a permanent link that is what I've been sending. So if for some reason you cannot come in person, I will count your attendance if you tune in on Zoom. I strongly prefer that you be here because it's way easier for you to ask questions and interact and such. But for instance, somebody's waiting for a contractor and that was not expected, but I'm damned if I'm going to bring somebody in to sit in a classroom when they need something done to their house. So I think from here on, I think except for exams, which I will try to insist that we do here as much as possible. I'll give you the option of attending via Zoom as long as you type something in the chat box so that it records your attendance. There's just too many people who have this issue, me included. So, all right, a couple of things then. If you are in the lab, we are having lab regularly scheduled. Half of the Wednesday lab will meet today. That's A through HA. And we'll go to City Park after we meet in Biology 216 to go over what we're going to do so that you're not out there flailing around and falling into the bayou or something. We'll do that for both of the lab sections. Half of the lab section on Friday will meet, that's A through K. The other half of the lab sections will meet to do that same exercise next week. And those of you who are meeting today will not have anything scheduled for next week. Those of you who are meeting next week do not have an activity scheduled for today. All right. So, oh yeah, the other thing is we are in that lab, we are comparing data that we're collecting this year to data that previous classes have collected. You will not get data to analyze until all 4 of the measurements have been done this semester. So you won't even get anything to analyze until a week from Friday, regardless of which section you're in. Okay, we got to compile all that stuff because when you do a statistical analysis, you can't compare one thing to one other thing. You have to compare a sample that is more than one sample. Okay, you have to have like sample size of like 2 at a minimum, and we're going to have a sample size of 4. So don't sweat what we're going to do today, and all through next week is just go and collect that data. We'll compile it, we'll send it out after a week from Friday's data has been collected. Okay, we had a number of issues with the exam. Respondents did not behave well for some people. And some people did not have a reliable connection. There are some people who still need to have that exam scheduled. So I have a key that I will post, but not until everybody's finished. This has been pretty difficult for a lot of people.
Excuse me, Professor. I got a question.
Yeah.
I was barely here last week. I just want to know, do we have a lab?
We did not have lab last
Okay,
week, no.
good. I'm like, I was freaking out. Okay, thank you. Cool.
Nope. Okay, so yeah, if you're in the lab, you are good. Now, one more thing about the lab. We have lab reports from the first exercise. I'd like to get them in by the end of business a week from Friday. If you have, some of you sent me stuff already. That's fine. It's just that with all of the other emails, I'm still working through them. So, you know, we will catch up, and you will get a grade on your first lab report as soon as I can make sure that I have everybody's lab reports. Yes. You can, you know what, actually, I prefer that you hand it in because I can just scroll on it. So if I see you today and you have it ready, you can just hand it to me today. If I see you Friday and you will have it ready Friday, that's fine too. If you've emailed, as some of you have, I haven't lost those emails. There's just hundreds and hundreds and hundreds of emails from you. So be a little patient with that, and we'll get this done. Okay. And hopefully that next storm turns north, as it should. Okay, so do we have any questions before we continue on? Yes, we have a question. We meet, if you are in the lab, we will meet in biology 216 before we go to City Park. Because I need you to be clear on what we're doing. It will take us a little time to get out there and do what we need to do. And I want to go over it so that you are not bumbling around and wasting a lot of time. All right, so we will meet in 216 before we go. Regardless of when you're doing this lab, we're meeting in 216. Okay. We will tell you about the arrangements for the following lab, the ensuing lab, as time goes on, right? But this lab, where we go to City Park and do community exercise, meet in 216 before you go to City Park. I need to talk to you about parking for one thing. People get tickets out there, and don't know if you knew that. So there are safe places to park and not safe places to park. Anything else? Okay. Well, let's start this next set of topics. And I don't know why I have two slides of this slide. That's kind of weird. All right, so we're still on populations. We're moving through our topics. The last presentation focused on the distribution of species and their dispersion patterns. Okay, and as I mentioned, the lab did one way to measure dispersion patterns, which is to look at the nearest neighbors and the distances between them. And we covered this kind of simpler, easier to comprehend way, simpler to explain way in our presentation, which is to count the number of individuals in an area of given size and do a whole series of those areas and see what the abundance within those areas is. And does it match a random Poisson distribution or not? If it's regular, then you'll have pretty much exactly the same number of individuals in each of those units that you sampled. If it's aggregated, you'll have a lot of those samples that have like 0 and some that have like 100, 200. One of the reasons that we did the dispersion exercise this year that we did was because the one that we did last year had exactly that problem. We had like 60, 80, 200 individuals within our quadrats. That was a little bit extreme. So I switched it up and we measured dispersion on something else. But now, let me see, did I hit that? I think no, okay. Now we're going to switch to a couple of new characteristics of populations, and that is abundance and density. Okay, abundance is kind of a qualitative measure of the number of individuals that a population has. It's whether it's common or whether it's rare. And density, on the other hand, is a quantitative measurement of the number of individuals in a unit of area. So commonness and rarity can be a little bit subjective, but density is not subjective. Density is quantitative. It has numbers attached to it that are not arguable. So we'll look at these two things today, and these are a little bit on this will be easy for us to get through today. It's a good way to resume our class. And then as we move on, we'll start to talk about age distribution and growth rates. And we'll spend most of the time talking about populations on those two things, trying to link them up. So abundance. Abundance is important for us in ecology because it is linked to the persistence of populations in evolutionary time. Common species tend to survive. Hello? Oh, let me. I think I didn't mute my phone. Let me do that. Common species tend to survive. Rare species are at risk of going extinct. Okay, so if you have a large population, there are several things that you have immunity to. Not complete immunity, but relative immunity. For one thing, there's something called environmental stochasticity, and that's random fluctuations in the environment, like the one that we had on August 29th. Okay. We also have droughts. That's kind of a random fluctuation. And so if you are adapted to a particular set of environmental conditions, those are not fixed in stone. Those vary randomly sometimes. So you can get a particularly dry year, a particularly wet year, and that may hurt you. It may cut your ability to reproduce. It may actually kill some of the members of your species. But if you have a large population, the effects are likely to be less than if you have a small population. There's also demographic stochasticity. Demographic stochasticity relates to patterns of births and deaths. Now, what you're going to see as we go on for the next few lectures is that we think of populations as having kind of an average birth rate or death rate, but really, that's not quite right. We have fluctuations in those things. And sometimes organisms can't find a mate. And so although the average birth rate per year might be 2, well, that year it was 0 for a lot of those individuals, and the birth rate on average is a lot less than 2. Okay, and you might have a fire go through and kill a lot of the adults, and the death rate then skyrockets. So that's kind of demographic stochasticity. Environmental stochasticity, think of as more affecting as well, it can affect both birth and death rates, but demographic stochasticity is not always the same thing as environmental stochasticity. It can be like the numbers of individuals in a particular area just prevent them from finding mates, or in a particularly good year where you have a lot of individuals packed together, everybody gets to mate, and everybody gets to mate multiple times, and so you have extra births. So environmental stochasticity and demographic stochasticity are often related, but they're not exactly the same. Demographic stochasticity can be due to the processes within the population itself. Okay, finally, genetic load is a big deal for small populations. In small populations, where you have deleterious alleles, they can become expressed at a much higher frequency in small populations. And they tend to be masked in big populations much more due to heterozygosity. But if you have a small population, then matings within that small population tend to put those deleterious alleles together at times. And when you have homozygosity for these deleterious alleles, well, that causes, at the very least, subfertility. It could be lethal. So having a large population keeps those bad alleles masked by heterozygosity. They're not expressed a lot, and the population doesn't have a big load of deleterious traits. Small populations tend to do this, and they can become genetically inviable, even if the environment is good. Okay, so this is a critical consideration in population biology. This is why there were translocations of panthers into Florida to raise the Florida panther population, because they were very much at risk of accumulating deleterious mutations, and those were being expressed in some of the individuals in the population. It was clear to wildlife biologists that the population was beginning to suffer genetically from its small size. So they transplanted some individuals from elsewhere, and that population is reasonably healthy at the moment. But that's the key thing. Population size, that is the abundance, is really important in conservation biology. So on average, local abundance is inversely related to the size of organisms. And here you see a whole series of studies that examine this. And so on the top left here, we have herbivorous mammals. So this is a very uniform set of organisms. That's this one over here for those of you at home. And notice that it's a really strong relationship. Really strong. So large animals tend to be rarer. Kind of end of story. This is not just within this group, though. This is general throughout all of biology. So here's plants, and here are annual herbs on the right, and then perennial herbs, which tend to be a bit larger, and then trees. And notice the cloud of, they didn't actually plot points themselves. They just plotted a sort of a blob. But you can see that it has exactly the same relationship. So annual plants tend to be smaller and more numerous than large plants. And the outlier down here at the bottom is the redwood that's found in California. And these are some of the largest organisms on Earth. They're very rare, partly because they are so large. You just can't pack that many redwood trees into a given area. On the other hand, you can have a whole bunch of dandelions, or in this case, duckweeds. Those of you in the lab, you'll appreciate duckweeds in a couple of weeks. And then finally, here's a much more general plot on the bottom left of all animals. And so we have aquatic invertebrates in red. We have, terrestrial invertebrates in blue. Okay, vertebrates, these are the poikilotherms in green. So we're talking about lizards, snakes, turtles. And then here are your birds and mammals, birds in yellow and mammals in red. And it's the same relationship. Large animals are rare animals. Large plants are rare plants. Yes, question. Okay, the question is, what are each of the points? What do each of the points represent? These are selected species, average sizes, and average densities. Okay, so here is a very large mammal down here. These are elephants. and notice that their density is relatively low. Okay, and up here, these are probably, I don't know, something like Collembola, these tiny, tiny little insects. Okay, and they are really tiny, but they're very abundant. Okay, yes. Yes, so this actually relates abundance to local density. All right, so that's what we're looking at. Local density is going to be here on the y-axis for all three of these graphs, and the mean body size in grams or kilograms, as the case may be, is on the x-axis. So this shows you the relationship between abundance and local density. Between body size and local density. So the take-home message here is that small organisms are more buffered from environmental, demographic, stochasticity, and genetic load than large animals. Large animals are very, very vulnerable to these things. That is why a lot of conservation biology focuses on those large animals and plants, because if you make conditions that help preserve those populations, almost by definition, you're establishing conditions that help the smaller ones that live in the same area. So that's why conservation biology frequently focuses on flagship species. It's not a reason to ignore the small species, though. They have their own special needs, and sometimes those are not addressed simply by doing what's good for, say, a redwood tree. All right, now, there are some real issues in applying the ideas of ecology to conservation of species. And one of the things that you need to know is not just how a species is rare, not just whether a species is rare, but how it is rare. So Deborah Rabinowitz devised a scheme to help translate ecological factors that influence abundance into something that conservation managers could use to guide their decisions. And she decided that what she laid out was a scheme in which there are sort of three key features. One is the geographic range, of course, which we talked about last time. The tolerances of those species. So how broad or narrow are their tolerances? Do they have a broad niche, or do they have really specific requirements? And then finally, what is the local population size like? So you can be rare in all of these ways. Now, populations that are least threatened have large geographic ranges, have broad tolerances, that is, they are generalists, and they have large local populations. If you are not one of those things, then you are vulnerable in some way. But you'll notice that exactly which way a particular species might be vulnerable to extinction differs for different species. A species that has small local populations would have different considerations than one that has a restricted geographic range, but might have a whole bunch of individuals within that range. So this is a really helpful way for conservationists to try to understand what the needs of species are and to try to address them. So here's an example. Now, all of these species are not going to be terribly familiar to you, and this will not help you visualize for all of them, but we'll pick out some that may help you visualize what's going on. So here at the top, we have some species that are really not vulnerable to extinction. The house sparrow is found pretty much worldwide, extensive geographic range. The house sparrow eats pretty much anything, and it has huge populations. And those of you who've ever had a lawn to take care of will know that dandelions are found pretty much everywhere. You cannot kill them, and they grow under most conditions, and there are a lot of them. So this is an example of species that have all three of these factors working for them. Okay, everything else has some type of rarity that might make it vulnerable. And here are some examples. So just below us, we have the Galapagos finch, medium finch, and it has a restricted geographic range. It's found only on a few islands on the Galapagos. Although there are many of them, if something were to happen to that island environment, that's a risk for extinction. Okay, the Monterey pine is a locally common tree that's found only on the coast of California in a range of about, I'd say 50 miles within 50 miles of the city of Monterey. So it's got a very restricted geographic range. It's common in that range, but if something were to happen to that local area, that species would be at risk of extinction. Okay, narrow habitat tolerances. Let's see, so the California gray whale ranges all the way from the Gulf of Alaska down into Mexico. It's got a huge geographic range. Okay, there are a lot of them, but it really has very strict requirements. And the key requirement for gray whales is actually the lagoons in Mexico where they give birth. That is actually the key narrow tolerance that they have. Okay, the Fremont cottonwood I know nothing about, so I'm not going to talk about that. Tigers, they have small local populations. Tigers are found from Siberia through China, Indonesia, and India and Pakistan and Iran. And that's probably about as far as they went, but that is most of East Asia. However, we have persecuted them to the point where they have tiny populations. There are only about 50 Amur tigers left in Siberia that we know of. There may be more because the Russians are actually trying very, very hard to help that species. So the grass fern, I have no idea what that even is. So here are some examples of species that have two of those factors and are at greater risk because of it. So the fish crow is a local species here in the Southeast. It's a restricted geographic range, and it's a fish crow. They're not generalists. They hang out on shorelines. So there are two reasons why that species could be at risk. Okay, the Haleakala silversword, that I know a lot about because that's one of the most common plants on the volcanoes of Hawaii, but it only occurs on the volcanoes of Hawaii, and it only occurs within a narrow elevational range. So it's got very specific requirements and very limited distribution. You get an eruption on some of those volcanoes and no more silver swords. Here are some that have restricted geographic range and small populations. Tasmanian devil is found only in Tasmania, and it's a top predator, so it has small populations because top predators have relatively few individuals. Okay, the Welwitschia plant in the Namib Desert is really kind of bizarre thing that looks like something out of Star Wars. This has actually a very good depiction of it. It's got this these huge leaves, maybe 20 feet long. And they just trail on the ground, on the sand. But it's only found in the Namib Desert, in the coastal dunes, and there are not very many of them worldwide. So northern spotted owl is an example of a species with relatively narrow tolerances and small populations. And the most vulnerable of all are those species that have all three of those things working against them, like the mountain gorilla, which is only now found in the mountains on the border of Rwanda and Congo, and only at mid-elevation cloud forests, and there just aren't many of them left. So it's important to understand how these things work for or against species. And in making conservation decisions, that is applying the principles of ecology to try to manage these populations, you have to know what is really the crucial thing for these populations. So if you're going to take conservation biology from Dr. Anthony, keep that in mind. It won't be the only thing that I'll ask you to keep in mind if you're going to take that course. You've already taken the course, you took it in the wrong order, sorry. Okay, let's move on to density. Okay, density is quantitative. It is abundance per unit area, always. Okay, and it's a function of local population dynamics, births, deaths, immigration, and emigration. And just a heads up, that's going to be the topic for our next presentation. It's called demography. Okay, but density determines whether you're going to have interactions like competition or aggression, where they're going to be able to find a mate. And so it also, you know, if you have a dense population, you're also likely to attract predators or parasites more than if you have a sparse population. So density is quite important in ecology. So we always talk about density in relation to a unit of area. So my example here is the number of elk per square kilometer. And, you know, pick something. Dandelions per square meter. Aphids per centimeter of leaf area. Those are all measures of density. So we'll finish this up by talking a little bit about how the importance of density and then how we measure it. So density depends on the fit between the environment and your needs, which we call the niche, right? The niche is the collection of needs and tolerances that a species have that allow it to persist and, most importantly, reproduce. The better the fit between the environment and your niche, the greater the density a population can have. So if you have some of your needs fulfilled to some extent, well, yeah, you can probably persist and maybe even reproduce there. But if all of them are fulfilled, you can have very dense populations. So organisms avoid areas that are not fulfilling their niche requirements. They migrate to areas that do fulfill them. And within those areas, they seek out the ones that are more favorable. Okay, and if you take animal behavior, we'll explain how they assert themselves in those environments. We're not going to do that this time. But they will either die or leave unfavorable environments in favor of more favorable ones. So let's think about how we envision the niche and how it relates to population density. So here we have a little cartoon. We've got two resources, resource one and resource two. Those resources are not defined. It's irrelevant what they are. But what we've done then is defined an area, a combination of these two resources that allow a population to grow and to reproduce. So this outer boundary here defines the niche. Everywhere on that line, which is this line here, everywhere on that line, if you are exactly on that line, you can survive. If you're outside that line, you die. If you're inside that line, not only can you survive and reproduce. Okay, but notice that it's kind of a bullseye. It's a target. And more favorable conditions are found within the niche. I've only sketched one bullseye. Sometimes species have combinations, more than one set of combinations that are really optimal. But in this case, we've got a set of pretty good conditions defined by the gray shading here. Okay, so these are favorable. This is, you can reproduce in the white area inside the boundary, but this is a more favorable area within the gray area. So you reproduce at a higher rate. Well, who wouldn't want to reproduce at a higher rate? So animals or plants seek out these more favorable areas. And you have higher densities in the gray area than you do in the white area. So this is our more favorable area. This is our somewhat less favorable area. You can reproduce in both, but you reproduce better here. So more individuals seek out the gray area. Okay, and here's your optimal set of conditions. Not only can you survive, you can reproduce at the highest possible rate. You maximize your reproduction. So the dark gray here is the optimal conditions. The population grows at the maximum possible rate. So this is how we think of the niche in the abstract. So the question then is, well, do organisms really do this? Is this what we see when we look at nature? And the answer is yes. Here's the actual feeding niche of a gnatcatcher in California. And you can see that this is the height above ground on the y-axis and the length of the prey. So there's the size. And these guys have their maximal energy intake right here at a prey length of about 3 millimeters, so relatively small, and about 3 meters above ground. So this little area in here is the bullseye. This is where they get the highest rate of food intake, of energy intake, and that if they can forage in those areas on those things, they will have more babies than conspecifics that are eating larger things at the same height above ground, or the same kind of prey higher up. So these rates are lower. These rates of reproduction would be lower than here. So this is a larger prey at the same elevation, or elevation, the same height above ground. There are just not as many of them. So if they focus on those big ones, their rate of energy intake is going to go down because they don't find many. And up here, they're higher above ground looking for the same type of prey. I have no idea why they would be less successful, but they are. The point is that our imaginary niche here, which we can think of in the abstract, actually does match what organisms do in nature. So you do have a correspondence between environmental conditions, the niche requirements, and local density. It actually works that way. Now, last thing, how do we deal with measuring density? Okay. Well, if you have a sessile organism, that is one that doesn't move around, conveniently plants are sessile, you can do something like a quadrat. And remember, we talked about basically sampling units when we talked about dispersion. Well, that's a quadrat. So you put down something like this frame that's depicted in this slide, and then you count the number of individuals inside the frame. Okay, that's a measure of density. Okay, so that's really pretty easy, and it's reproducible. You have to maintain the same unit of area. And for those of you who are in the lab today, we're going to use a particular unit of area in our study. And we count those organisms in each of those units, and we take the mean density then. If you get a mean density of all of those areas. You can then multiply by the area that you're interested in and sort of estimate what the local population size is. And this is how we proceed in almost all of ecology. We almost never have the opportunity to actually count every individual in a population. We almost always are estimating. And so if you've got an average, then you also have to have a standard deviation so that you have some idea of how variable it is. But this is how we proceed. Okay, and it's relatively simple to do sessile organisms. To do mobile organisms is a bit harder. Here are some examples of how we measure density in these guys. The problem with this is that they move around. So you can't tell if you're double counting. So you have to actually mark individuals and you have to then try to see them again. So here's some examples of marking individuals. Here's an elaborately marked deer in the middle, but you see you got bird bands on the top right, and on the bottom right, you have paint marks on a monarch butterfly. So you simply need to mark each individual uniquely and let them go. And then you come back at a later date and you ask, if I see like 100 of these individuals, how many of them are marked? And if you know the number that you marked initially, and you know the proportion that are marked in your second sample, you can then estimate the sample size. Because when you first went out and marked some, you took the entire unmarked population and you took a fraction of them and marked them. So maybe you marked 10 out of 100. You may not know it's 100, but you know that you marked 10. If you come back later and you see 20 individuals, and you see that 2 of those individuals are marked, well, that's 10%. So 10% of your sample was marked. That means that you marked about 10% of the original population the first time. So it's just very simple algebra. And there it is. And here's an example. This is an example, the total vast population in Bayou St. John. Okay, so we don't know that, but we're gonna mark some. So we're gonna go out and take a net and we're gonna mark 100 bass. And maybe all we're gonna do is say, cut a little notch out of the fin. Okay, and then we're gonna put them back in the bayou. And we're gonna go away. And if our marking doesn't harm anybody, this is an important assumption. If our marking doesn't harm anybody and we come back later and we see that we capture 500 and 10 of them are marked, well, okay. 10 out of 500 ought to equal 100 out of whatever this number is. So 10 out of 500 should equal 100 out of the number of bass in Bayou St. John. It's just simple algebra. Just rearrange it. So 10n equals 50,000, and that means that n equals 5,000. There's approximately 5,000 bass in Bayou St. John. This is called the Lincoln Index, and it's one of the most fundamental ways of measuring density in ecology. It's very, very simple, but as you can see, it has some assumptions. It assumes that your marks don't hurt anybody. If you mark an individual that makes them really visible to predators, well, then you're going to capture fewer in your second sample than you would have otherwise. And that's going to skew your estimate. Also, if you use, say, a felt tip marker, and that's toxic, oh dear, well, that's going to skew your estimates also. So, we have some assumptions with this, but this is a very simple and very, very useful tool for estimating densities. And what this also assumes, though, is that there are no births or deaths in the population that would affect your estimate. So you can't mark a population of, say, something like fish 1 year and then come back another year and expect to have an accurate estimate. You can do that, but it's much more complicated, and there are a lot of things that you have to take into account if you do something like that. If you sample one time and then reasonably quickly get a second sample, though, you can pretty much assume that those processes are not going to affect your estimates. And as long as you didn't do anything to those marked individuals that caused them to die, then you can get a reasonably accurate estimate. And I think you can see what would happen if your mark did cause, say, half of your marked individuals to die. You'd only capture 5 here instead of the 10, and that would be 5 n equals 50,000. So you'd estimate a larger population than is actually there. So if you do something that affects the survival of your marked individuals, you'll get an inflated estimate of the population size. Okay. That's really about it for today. I'm not going to say anything more about this because we have a lot to do when we talk about demography and then population dynamics. So unless we have any questions, I will close this out for the day and see those of you in lab at 1 o'clock. All right. See y'all later.